Okay, so we were discussing what happens to Lac Operon under different conditions. So I was telling you about when we make it mutant, the operator becomes mutant, what happens? So operon will be constitutively expressed because operator will not be able to bind to, or the repressor protein will not be able to bind to the operator because it is mutant. So the operon will be constitutively expressed irrespective whether lactose is present or lactose is absent. Okay. Let's say what happens when we make it partially diploid with a wild type Lac operator. That means the original Lac operator is mutant, shown over here as Lac O superscript minus mutant. And what you have done, you have added a piece of DNA containing the operator region and you have incorporated it, you have added it to the bacterial cell. Please mute yourself off. जिसका माइक चालू है वो बंद कर दे. So we have made the cell partially diploid for operator region. So what is going to happen? Okay, the operon will remain constitutively expressed. Unlike when we added Lac I, wild type Lac I, and made it partially diploid for Lac I, the wild type inducibility was restored here. But in this case, when you make it partially diploid for Lac O, the inducibility is not restored. The operon is still constitutively expressed because wild type inducibility is not restored because the operator is cis-acting. The operator is cis-acting. So I told you yesterday what is meant by cis-acting and what is meant by trans-acting. Let us understand that concept once again. So cis-acting sites are what type of sites and what are their characteristics? Let us try and understand. I'm sorry, there is something wrong with the connectivity over here. So cis-acting sites, what are their characteristics? These are the sites which function, please understand, by being recognized. They are called as cis-acting sites. These sites function by being recognized. So they are called as cis-acting sites. The other characteristic of cis-acting site is that they need to be physically contiguous to the site they control. They need to be present adjacent to the site they are controlling. Okay, physical closeness is necessary for a site to be cis-acting. Okay, and third, this cis-acting site, they remain unaffected by the presence of other alleles of the site. What does it mean? It means when you made it partially diploid by adding another LACO, wild type LACO, yeah, it's an allele of that gene. Okay, so you have added another allele, a wild type allele, so it remains unaffected. Addition of that wild type LACO doesn't restore the wild type inducibility in case of Lac Operon. So what does it mean? It remains unaffected. Cis-acting sites, they remain unaffected by the presence of other alleles of the site, or you can say gene also. Okay, so when you add a Lac O plus, a wild type Lac O, it doesn't really affect the status of the Lac Operon. It remains continuously constitutively expressed only. Vis-a-vis trans-acting site. What are their characteristics? So trans-acting sites, these are the sites or these are the genes which function by producing a diffusible product. Usually they code for something. That is usually a protein. Sometimes it can be RNA also. These sites are called as trans-acting. Cis-acting sites normally do not code for anything. Trans-acting sites, they code for either a protein. In majority of the case, it is a protein. Sometimes it can be an RNA also. The diffusible product, the protein or the RNA, can act on a relevant site present on same or different DNA molecule. So now probably you will understand when you make it partially diploid by adding a Lac I plus, a wild type Lac I, that diffusible product can act on relevant site. If it is the same site, that product repressor protein will work on it, will act on it, whether it is present on the same DNA molecule or it is present on a different DNA molecule. Okay, so you have your original thing is mutant here. Here it is mutant, let's say. Lac is mutant. Okay, but what you have done, you have made it partially diploid by adding a wild type Lac, right? So since it codes for a diffusible product, this product will be normal product. It will go and bind to the operator region here present. Okay, so the diffusible product can act on a relevant site present on same. It is not necessary that it should be present here only on this DNA, because it is being produced by this Lac. So not necessary that it should be present on the same DNA molecule. Even if it is present on some other DNA molecule, relevant site is present on some other DNA molecule, the diffusible product will go and act on it. Third characteristic is the presence of other allele can restore the originality. Okay, so originally when Lac gets mutant here, the operon was constitutively expressed. All the time it was expressing. But when you add other allele, which is wild type, okay, what does it do? It can restore the originality. Means the wild type inducibility is restored when you make it partially diploid for that particular gene, right? So this is very important. Please try and understand. I'll share this PowerPoint presentation to you in the classroom. Okay, so let us understand. This is the Lac operon now. You have a Lac gene, there is a promoter, there is an operator, and these are the three structural genes. Okay, and somewhere here, you have the terminator, transcription termination signal also, right? So what happens? Lac okay, what is its action? It is transacting. So we have just understood it that Lac codes for a diffusible product. So therefore it is transacting. Okay, and the product is what? The product is a repressor protein, right? Operator, what type of site it is? It is cis-acting because it functions by being recognized. And who recognizes it? Recognized by the repressor protein. It is recognized by the repressor protein. Promoter, okay, promoter region, this region. What type of site this is? Promoter is also cis-acting. Physical closeness to the structural gene that it is controlling is necessary. Okay, and it functions by being recognized. Who recognizes it? RNA polymerase. It functions by being recognized by RNA polymerase. It doesn't code for a diffusible product, it doesn't code for a protein, it doesn't code for a RNA. Let us understand terminator. What kind of site this is? This is again cis-acting. This terminator site is cis-acting. Why? Because it functions by being recognized. Who recognizes it? Once again, RNA polymerase. RNA polymerase recognizes it and brings about the termination of the transcription. It doesn't code for a protein, it doesn't code for a RNA, it functions by being recognized, and therefore it is also a cis-acting site. So I think I have amply made it clear the difference between cis-acting site and trans-acting sites, right? Now, today's topic is catabolite repression. Now, the operon, now you know, this Lac operon, it gets switched off in the absence of lactose, and it gets switched on in its presence. When lactose comes, it's pretty simple now. When the substrate comes, those enzymes will be synthesized. When the substrate is not there, there is no point synthesizing those enzymes. But there is a twist now. What will happen when glucose is also present along with the lactose? So you have presence of lactose, but along with lactose, you have glucose also present. So what is going to happen? Will it be on? Will it remain on, switched on because lactose is present, or will it get switched off? This is the question that we will be answering today. Please understand and note that glucose is a much preferred source, ready source of energy for the bacteria, because lactose will have to be taken up and it has to be catabolized into glucose and galactose, and then the glucose will be utilized. But when glucose is available ready-made, it is a preferred source and ready source of energy. So in presence of two sugars, when both lactose and glucose are present, the operon should be switched off, because it is utilizing glucose. Then what is the point, synthesizing enzymes which are required for the uptake and catabolism of lactose when it is utilizing glucose? Okay, so there is a twist now. When both the sugars are present, the operon gets switched off. The operon gets switched off. This switching off of Lac operon in presence of glucose is called as catabolite repression. So please remember this definition. What is meant by catabolite repression? The switching off of Lac operon, switching off of Lac operon in the presence of glucose is called as catabolite repression. And this serves as an additional control mechanism to avoid wasteful expression. This is an additional control mechanism so that when both the sugars are present, when glucose comes into the medium, this operon can be switched off and avoid wasteful expression and use of resources, scanty resources. It is a part of global regulatory circuit. Means this kind of regulation is available in many other operons also. Okay, so in the physical class I keep giving this example. When you go to a hotel, there are switches inside the room. You can switch on the AC, you can switch on the fan, you can switch on the light, and there is one switch outside the room. So when you go out, what do you do? Instead of switching off all the switches inside, or by mistake, suppose if you have kept something on inside, by switching that switch off, which is located outside the room, basically what you are doing, you are switching off all the appliances within that room. So it is this kind of a, what you can say, regulation here. It is an additional regulation along with the usual regulation that is there. Now let us understand how does it happen. So what was found out is that glucose level and cyclic AMP level, they are related. And how are they related? They are inversely related. Cyclic AMP in the bacterial cells serves as a hunger signal, and it is related to the level of glucose by an inverse relationship. I have tried to show it over here. Cyclic AMP and glucose are, levels of these two are inversely related. What is meant by this? When the glucose is high, okay, cyclic AMP level is low, and vice versa. When the glucose is low, cyclic AMP level is high. Okay, this is what is meant by inverse relationship. Okay, now this cyclic AMP in the bacterial cell, it binds to a protein called catabolite gene expression protein, CAP. Cyclic AMP, it binds to a protein called catabolite gene expression protein. Okay, there seems to be some more problem with the internet connection here. Okay, this catabolite gene activator protein, CAP, it is a dimer of identical 22 kilodalton subunits. It's a dimer of identical 22 kilodalton subunits. And it has two binding sites. One site is there where cyclic AMP comes and bind. This is the cyclic AMP binding site. And there is another site which is the DNA binding site. With the help of that site, it will bind to the DNA. So it has two binding sites, one for cyclic AMP shown over here, and there is other site, with the help of that, it binds to the DNA. This cyclic AMP CAP complex, it binds to a region in the promoter and it stimulates transcription by a factor of 50. Presence of this cyclic AMP CAP complex at a particular region in the promoter of the Lac operon is necessary to, is necessary for efficient transcription. It stimulates, it facilitates transcription, and stimulate it by a factor of 50. In presence of cyclic AMP CAP, transcription takes place radially from the Lac promoter. Otherwise, it takes time and the frequency of transcription from the Lac promoter becomes very low when this cyclic AMP CAP is not there. Okay, so should I repeat it once again? I'm repeating it once again, that glucose and cyclic AMP, they are in inverse relationship. This is point number one. Cyclic AMP, it binds to a protein called catabolite gene activator protein, CAP in short. It's a dimer of identical 22 kilodalton subunit. It has got two binding sites, one for cyclic AMP, another for DNA. And this cyclic AMP CAP, with the help of that DNA binding site, binds to a region in the promoter and stimulate transcription by a factor of 50. Presence of cyclic AMP CAP in the Lac promoter is necessary for RNA polymerase to come and bind and efficiently initiate transcription. In the absence of these, RNA polymerase finds it difficult to identify this promoter, and the frequency of transcription from the Lac promoter becomes very, very low. Means you can say it is almost switched off. RNA polymerase finds it really difficult to identify this promoter. What is the reason? Let us understand that reason also. So this is how, these three proteins, they bind in the promoter region of the Lac Operon. Cyclic AMP CAP binds next to RNA polymerase binding site in the promoter, and it protects the region from -87 to -49. So minus, you understand, upstream. So this is the start point. These are the upstream sequences. So cyclic AMP CAP, it protects a region -87. This is -87. This is -87. So this is -87 and this is -49. Right? So cyclic AMP CMP, it binds to a region in the promoter. I have given you the nucleotide numbers, -87 to -49. This is -87 and this is -49. RNA pol, the one in blue, shown over here, RNA pol, it binds to the promoter between -48 to +5. So this is -48 up to 49. Cyclic AMP binds -48 to +5. Getting into the structural region, this is the start site. So it gets into the structural region. These are upstream sequences. Upstream region, this is downstream. So in the structural region, it extends about 5 base pair. So -48 to +5 is being protected by RNA polymerase. And you see the repressor protein, operator region. Where does it extend? It extends from -3 to +21. Okay, that means the operator, means repressor protein binding site, and the RNA polymerase binding site, they overlap. There is an overlap. It goes from -48 to +5, whereas repressor protein binding, it starts from -3. So there is an overlap of around 8 nucleotides, and therefore only when repressor is bound, RNA pol cannot bind, and when RNA pol is bound, repressor cannot bind, because there is a overlap over here. Seen over here also, there is an overlap here. There is an overlap of 8 nucleotides, whereas there is no overlap over here. So this presence is must for RNA polymerase to come and bind to this region. If this is not there, cyclic AMP, CAP alone cannot bind. CAP has to have cyclic AMP bound to it. Means it should be a complex of CAP cyclic AMP. Then only it can come and bind. CAP alone cannot bind. Okay. So when it is there, it is sitting over here, then only RNA pol can come and bind. And when RNA pol binds, repressor cannot bind because the two sites are overlapping. You can understand it by memorizing these. By this figure also, from this figure also, you can understand that there is an overlap here, right? What is the necessity? Why this happens? Okay. cyclic AMP CAP binding is necessary for RNA pol to bind to Lac promoter, I told you earlier. When glucose is not there, okay, and because of the inverse relationship, when glucose is not there, cyclic AMP becomes high. When glucose is low, cyclic AMP goes up. So it binds to CAP, and in turn the complex cyclic AMP CAP binds to the promoter region and facilitate binding of RNA polymerase. Transcription begins. Okay, when glucose is present, okay, when glucose is there, cyclic AMP is going to be less because there is an inverse relationship. So since cyclic AMP is not there, it will not bind to CAP. And when CAP alone cannot bind to the designated region in the promoter. I told you CAP by itself cannot bind. It requires a complex. It requires binding of cyclic AMP, and then the complex only can go and bind. So RNA pol therefore cannot bind. No transcription. What is the reason? Why so? The reason is that Lac promoter is a weak promoter. Please remember, I told you earlier also what is meant by weak promoter and what is meant by strong promoter. What is meant by weak promoter? You know, we have seen that those -10, -35 consensus sequences. So in a standard promoter, TATAAT is the -10 consensus sequence, and TTGACA is the -35 consensus sequence. And if they are there as it is, without substitution, without deviation, then that promoter is called as a strong promoter. And when you have multiple substitution, that promoter progressively gets weaker and weaker. And you see the Lac promoter, the -10 sequence, TATGTT. The nucleotide in red, they are the substitutes. You know, originally it should be TAAT, but you have TGTT. Okay, so there are multiple substitutions. Same is the case in -35 consensus sequence in the Lac promoter. You see the sequence. What is required is GACA, you have ACAC. So there are multiple substitutions in the -10 and -35 consensus sequences, and as a result of that, Lac promoter is called as a weak promoter. So when the promoter is weak, RNA polymerase always find it difficult to recognize these kind of weak promoters. RNA polymerase requires some additional help to identify this promoter, and the additional help in this particular case is that binding of cyclic AMP CAP complex to that particular designated region. What is that designated region? So I told you that designated region is, you know, -87 to -49. This is the region, -87 to -49. Okay, unless cyclic AMP CAP binds here, RNA polymerase will find it difficult to bind to its place over here. So when it comes there, when glucose is not there, cyclic AMP is high, it binds to CAP, and cyclic AMP CAP comes and sits over here. And when lactose comes into the medium, you know, RNA polymerase can come and bind here, because presence of lactose induces a conformational change in the repressor protein, and repressor protein cannot come and bind to the operator region. So we have seen it in yesterday's lecture. So the reason now you know that Lac promoter is a weak promoter. Why it is called weak? So please compare these two sequences, consensus sequences which are there present in the standard promoter and the consensus sequences which are present in the Lac promoter. So now we'll have to, for efficient expression of Lac Operon, okay, our definition is now going to change. When lactose is absent, you say the operon is switched off. Fine. When lactose is present, you say the operon is switched on. It is partially correct. It is not totally correct. It is partially correct. When lactose is present and glucose is also there, the operon gets switched off. The operon gets switched off. This is what is called as catabolite repression. When lactose is present and glucose is absent, then the operon is switched on. Then the operon is switched on. Please understand, please remember this. So what you can say, for efficient expression of Lac operon, what do you require? You require presence of not only lactose, but you also must ensure that glucose is absent. For efficient, I'm repeating, for efficient expression of Lac operon, what do you require? You require presence of lactose, right? But you also require absence of glucose. Then you can say the operon is efficiently expressed. And therefore I said, when lactose is present, the operon is switched on, it is only partly correct, because you need to specify when lactose is present, glucose is absent. Then only you can say that operon is efficiently expressed, the operon is fully switched on. When lactose is present and glucose is also present, then the operon will get switched off. This phenomenon is called as the catabolite repression. So the meeting will end anytime now. I will be sharing this. Thank you very much. I'll be sharing this PPT in your classroom this afternoon. And I will have to record the video again because this video has not come properly because of the interruption in the